today I'm going to be talking about how to access and analyze neuroimaging data from online databases. Now, this is going to be a little bit superficial because we'll only be covering a survey of some of the options that are out there. And for now, I'm going to cover a few of the main ones that are out there. For example, Human Connectome Project, the Alzheimer's Disease Neuroimaging Database. And I hope that some of these might be of interest to you. And of course, if you want to know more about how to analyze them in more detail, you can always contact me personally and we can set up a meeting for that. So some of the trends that have led to where we are today. The first one is called open science. This has picked up a lot of steam in the past, let's say 10 to 15 years. And the way I'm going to define that for today is transparency and reproducibility. In other words, we all know that if you write a methods section for a journal publication, the method should be detailed enough so that anybody could, in theory, replicate all of your steps and reproduce the same results that you have. And reproducibility means that you can get the same results that somebody else did in their experiment. A closely related topic is replication, which means, are you able to get more or less the same effect even though you're using an independent data set? And these two trends have picked up, like I said, a lot of popularity in the past 5 to 10 years. There's also been advances in technology, both in scanner technology, also in supercomputing. And so a lot of what we want to do as far as analyzing structural data, diffusion data, fMRI data has become much easier, especially at scale, using supercomputing clusters. We're very fortunate here at the University of Michigan. We have a fantastic supercomputing cluster called Great Lakes, if you've ever used that, and also some very good support staff as well. Now, this has all led to a growing use of large databases, both hosting lots of data online and also the use of data for publication purposes as well. So like I said, I'll be giving you a survey of some of these larger databases and how they can be used. And to give you some history, I think that a lot of this was inspired by the Human Genome Project, which I'm sure a lot of you are familiar with. Formally, it started in about 1990. It was a worldwide initiative, gathered data from many different people, many different countries, in an attempt to sequence the human genome. So as you also probably know, the human genome is composed of different kinds of nucleotide bases. And the idea of this project was to try to form what is the most regular kind of sequences you would see in the population, and what are some sequences that might indicate the onset of certain kinds of different physical and neurodegenerative disorders. So this was formally concluded in around 2003. Over the next decade, they were still filling in some gaps, but more or less they have a pretty complete sequence of the human genome. This in turn, I would say, inspired something in 2013 called the Brain Initiative. So this was an initiative to try to provide more resources, more tools, and also more databases for people to advance brain imaging research, with a focus on trying to identify, prevent, and cure disorders of the brain. So this involves things like connectomes as far as structural data. So for example, in the bottom left here, you can see white matter tracts, which are color-coded according to their main orthogonal directionality. And you also have something on the bottom right indicating functional connectivity, which strictly speaking might not be connectivity. It's more correlations between the bold signal between different regions of the brain. But both of these can be combined to try to give a fuller picture of, again, what does a typical connectome look like and when might it go awry and show up in different kinds of subpopulations. For example, people developing Alzheimer's, people with mild cognitive impairment, or people developing schizophrenia. So some of these databases have their genesis in one of the first attempts back in 1999 by Jack Van Horn and Michael Gazzaniga. This first attempt was called the fMRI Data Center, or fMRI DC. They were both working at Dartmouth at the time. They got in contact with some different leading neuroimaging journals at the time, and they said anybody who submits their manuscript, encourage them to also submit their data as well. The idea being people can try to replicate what they did. We also can build up a database and get kind of a running history of what's been already done. And other people might find it useful as well. Interestingly, at the time, there was a lot of pushback from people who were submitting their manuscripts to some of these journals. They felt like this might be some undue pressure on them. They felt as though providing all their data might lead to them getting scooped. As you know, some people gather a large data set and they try to spin multiple publications out of it. It's a perfectly normal, acceptable practice. And they might be worried that somebody could get the jump on them and publish ahead of when they had already analyzed the full data set to its fullest extent. But this was an important first step, and over the next decade, as some of the privacy issues also were somewhat abated, there was some standardization in how fMRI data was being stored. You see the rise of some different projects as well, such as the 1000 Functional Connectomes project in 2010 and the International Neuroimaging Data Initiative in 2012. These are all attempts to gather large amounts of data, and some of them focusing on specific modalities. So for example, the Functional Connectomes project is focused mostly on functional connectivity data. In that case, most of the time, people are in a resting state. You collect both signal when they're at rest, and you form these correlations between different regions of the brain. More recently, something called openneuro.org, which had its origins in openfMRI.org, has also been much more widely used, and I find it very useful both for pedagogical purposes and also for research purposes as well. That's one of them which I'll show you in a more practical sense later on. I'll show you how to actually interact with it, download the data, and also possibly analyze it as well. Now, a few of the much larger ones, these go on over a much larger time scale. You may have heard of some of these. There's the ABCD study, which has been collected across multiple sites across the country. One of them is actually here at the University of Michigan. Last I checked, and still ongoing, had about 12,000 subjects. There's also the Human Connectome project, which has about 1,200 subjects, which has gone around for 15 years or so. And the largest one of all that we know of worldwide is UK Biobank, which, again, last I checked, had about 40,000 subjects and counting. Now, not all of these are equal in the sense that some of these are much easier to access than others. For example, the Human Connectome project is relatively simple to sign up for, and they have different tiers of access. Some are much more readily available to share and to distribute, to publish. There's also a more restricted set focusing on subpopulations, which requires a special application, and there are much more tight restrictions on privacy. Something like ABCD can only be accessed, as far as I know, through the NIMH website, National Institutes of Mental Health, and requires a few steps as well. Lastly, UK Biobank requires a fee. It's somewhere in the thousands of dollars, last I heard. It's not that easy to access, but it does have the widest range of both functional and demographic data. Now compare this to the typical fMRI study, and this is something I want to run side by side as I'm talking about all of these, because historically, up until now, the typical fMRI study has been around, let's say, 20 to 30 subjects, around 25 so far. And, there's a useful comment here. UK Biobank is about 10,000 for 3 years. We have access in our group if anyone needs advice. Thank you so much for that. And I encourage anybody who is interested to contact Venk for more information. Now, the typical fMRI study these days has about an N of 25. And so there's a lot of questions about, is the typical fMRI study really useful anymore? Because there are statistical power issues which have come up recently, which have actually been known about for quite some time. And some people are saying, shouldn't we just replace all of this with simply collecting or downloading samples from larger databases? And I'll be talking about that as well. So the first demonstration that I'll show you is accessing databases through something called LONI, or the Laboratory of Neuroimaging. It's a data management site through the University of Southern California. And through this, you can directly apply for access to many studies. So for example, there's the Alzheimer's Disease Neuroimaging Initiative. There's ABIDE, which focuses on subjects on the autism spectrum, the Mayo Clinic Study of Aging, and other ones as well. And so what I would like to do is break out from my slides here, show you my screen, and give you a brief rundown of how this might work. Now, just a heads up, I'm going to show you ADNI at a very superficial level because unfortunately, they do not want me to show subject IDs. But I'll show you the portal, I'll show you some of how to get it set up, and how a typical image search is done. All right. So to start with, we have this website. Again, this is through ida.loni.usc. I'm going to hide this for now. All right. And so through this, and I'm already logged in here, but you can see there are many different studies you can apply to. Through this, I only have access to ADNI. So the entire application process means you need to formally submit a request. You need to specify who is going to be allowed access to it in part of your group, what it's going to be used for, and of course, if you do any publications, they want you to both document it and also acknowledge them as well. So I'm already logged in. If I click on ADNI, for example, it should give you a display of certain features. Let's take a little bit. Instead, I'll just go to my account and give you a picture of what this might look like from the inside. So I have PI status. I've applied for basically just having access to this particular data set. And if I go to something like search and download, I go to, let's say, advanced image search. And again, without getting into actual subject IDs, for example, you can see here there are many different phases of this Alzheimer's disease neuroimaging initiative. So this includes both controls. It includes people with mild cognitive impairment. Some might call it the prodromal stage of Alzheimer's, and also people who have been diagnosed as having dementia. Yeah. Formally, Alzheimer's, you need to do an autopsy, but basically they have people who they're pretty sure have some kind of dementia that's indicative of Alzheimer's. And so through this, you can access many different stages of these different phases. Now, one thing I do want to point out is ADNI has been around for a long time. These phases cover different years, and they also probably cover different scanning protocols. This gets into a separate topic called scanner harmonization. How do you combine data from different, say, phases, different scanner sites? That's another topic I'm not going to go too much into, but just be aware of that. And so from here, you have access to different modalities: MRI, DTI, diffusion imaging, PET imaging, fMRI. And most of these, I should point out, there isn't a lot of task-based data with this particular database, but you do have access to a pretty rich array of structural images. That includes diffusion, it includes white matter, gray matter scans, it's a typical T1 weighted anatomical scan. Also things like white matter hypointensities, you can process those through FLAIR images as well. So essentially, you just check all the boxes according to whatever specific subtype that you're interested in, whatever specific phase you're interested in, and then if you click on search, it'll show you those specific subjects and you can download them. Again, just due to time constraints and also due to the public nature of this recording, unfortunately for this website, can't show you those particular subject IDs, but this gives you a broad overview of what's possible through LONI and also through the ADNI website. All right. So going back to my slides for now, I'm going to be covering next one called the NIMH Data Archive, or NDA, which also might be of use to you. So the first one, ADNI, again, contains more clinical scans, and also the NDA as well gives you access to many different repositories. For example, ABCD, which is one that I mentioned earlier, the Yale Autism Center for Excellence, and many more as well. Now, this is a little bit more restricted. It requires a login.gov account or an ERA Commons account. And it also requires that whichever institution you're affiliated with has FWA, or Federal Wise Assurance. So this is a brief picture of what it looks like. So for example, once you have your login.gov account and you go into NDA, you can request access to something like the NIMH data archive, which, like I said, gives you more access to many different repositories as well, depending on your needs. You can see some pending requests. You can see some other ones that have already been terminated, for example, other ones that I've worked on. Now, it is rather involved, the process. You have to submit a data access request by clicking on that button in the previous screen that I showed you. This will generate a data use certificate, colloquially known as a DUC. And like I said, it requires federal-wide assurance. University of Michigan does have it, for example, but if you're working at a smaller institution, private liberal arts college, for example, I can't guarantee that you do have FWA. So it is relatively restricted. It also requires a research-related need to access the data. And I should also point out that they do have pretty strict requirements about, again, privacy, where you're going to store it. And they also want you to delete it pretty much as soon as it's finished, whatever the purpose was, whether it's for research purposes, whether it's for pedagogical purposes. They basically want you to delete all traces of it once your timeline is done. So just a few more snapshots of what this might look like. So this is your DAR, data access request. So for example, you provide your institutional sponsorship at the very end of it. You give some brief demonstrations. For example, what exactly is the research purpose? What is the data access plan? For example, I say I'm the lead recipient. I'm only going to be used for demonstration purposes. At no point will the user IDs or sensitive information be displayed. Any figures will be de-identified, group averages, and so on. And also things like how it's going to be stored, data deletion program, things like that. So these are very important. I mean, it can feel like a lot of work and very tedious to set up some of these access requests, but there's good reasons for it. They're very tightly concerned about privacy, about HIPAA requirements, and so on, and we need to take it seriously as well. Lastly, I'm not giving a list of all the different people involved, but you will need a co-signing official. So this might be somebody, let's say, involved in submitting your grants, your R01s. Anybody involved in grants, for example, could be used as a co-signing officer. Essentially, it's somebody who vouches for you as an institutional official. So you just search within your institution for somebody, usually somebody that you probably already know and already have worked with. It should come up here, and we'll simply send them an email, and then they'll need to fill out the rest. So that's the NDA. I'm going to move on to something where I will be able to give you kind of a fuller treatment of both access and downloading. The Human Connectome project has, again, been around for quite a while. It's a collaboration between USC and Massachusetts General Hospital. And it contains structural and functional data. And compared to a lot of the other other large databases, quite a wide range of different functional tasks. Also both resting state, task-based data from about 1,200 subjects. Another reason that the Human Connectome project is pretty popular in the literature, they use specialized acquisition protocols, which a lot of other people like to adapt for their own research purposes. So HCP has a pretty well-known resting state acquisition sequence, and I believe they use Siemens scanners. I'm not entirely sure. But essentially, if you use the same scanners they do, or you want to adapt them to your scanner, it's a pretty simple way to do that. It's on their website. You're able to access it. And that's a pretty good resource for the community. Now, here's what the typical interface would look like. I'm going to show you just simply downloading a single subject from the HCP-Young Adult repository. You'll notice there's this download from ConnectomeDB. It does require an additional download add-on, which facilitates the downloading of the data through this particular system. So I'm going to break out of my slides, give you a brief demonstration of what that looks like. All right, so here is the Human Connectome project. The actual landing page is here, but we're going to focus on the young adult HCP. And over here, again, you do need to sign up for an account, relatively easy. I'm not going to go through all of that just because, it might take some time. But here, once you go to get data and you've downloaded this ConnectomeDB, which is you can also access through the website, you click on that particular button and you would have access to a few of these different studies within the young adult repository. So for example, they have 1,200 subjects. It gives you a brief description of all of them. So things like structural images, resting state, task-based, diffusion, so on and so forth. And then if you click on this drop-down menu to the right, you can say look for just subjects with a certain field strength, 3T, 7T. You can just download behavior data. It includes a full battery of things like demographic information, for example. Or for demonstration purposes, I'll focus on a single subject. And here notice that they have different means, different levels of processing that you can download. So for example, you have the structural data for this single subject, which has already been pre-processed. You might say, what exactly does that entail? Well, it includes things like, has the data already been segmented? Has it already been run through certain pipelines which generate things like inflated surfaces, for example, which can be useful for further functional processing and visualization. Also, for example, resting state, which has been denoised, for example. You could also download that as well if you want to use that for research purposes, teaching purposes, just for your own learning. For now, I'm going to focus on this first one right here, this structural preprocessed. What it would look like is you click on Queue for download. You could select multiple ones. I'm just going to select one, and then click on Download Packages. It gives you a summary of what's going to be downloaded. You click on Download Now, and then it launches this IBM Aspera Connect. And after about a minute or so, it will download it. So if I go to downloads over here, notice we have already downloaded it from the Human Connectome project. I'm going to simply unzip that. We'll take a brief look inside. What would this look like? Again, it's only a single subject, one modality, but you should get the sense of how this could be done for entire data sets and multiple modalities. If we look within something like T1W, some of these file names may not be familiar, and that's fine. What I want to point out is that within this, you see things like, the raw T1 data. You also see something called, ribbon, and also these other subfolders like FS average, for example, which contain different kinds of images that have a GII extension. This is called GIFTY. You may have heard of Nifty. This is GIFTY, which is a specialized file format that contains both surface-based and also volumetric data. Now, something about the Human Connectome project that you should also know about, and again, I'm not going to go through how to download this as well, but they have something called Workbench or WB View, which has been developed to work primarily with these kinds of files. So if I open up Workbench and I go to this directory I was just looking at, and let's say I want to look at any file, for example. Let me just pick something at random, one of these surface-based images. So this is a T1 weighted image that's already been processed, I believe, through FreeSurfer. It's generated these two-dimensional surfaces, which you can view. And if you had the time, you could also, let's say, process the functional data as well and project it onto the surface for better visualization and for higher fidelity for exactly where these functional data are localized within the grooves and the folds of the brain. So there are many more. Again, I encourage you to look at these. Again, I'm giving just a survey, brief overview of some of these different directories, of these different file formats and packages. But this is the basic setup of how you download from the Human Connectome project. And also just be aware that they prefer and they've developed a lot of their software and images to work with this particular visualization software called Workbench. So you will need to download that as well to interact with the data. All right. So that is the Human Connectome project in broad strokes. Something else I also wanted to, oops, don't want to do that. Connectome at UM used Philips scanners as well. Oh, thank you, Daria. I did not know that. So yes, they did use different scanners, not just Siemens like I thought. Okay. Before I move on from the Human Connectome project, I did want to point out one thing because it's a technique I've been using lately, which is how to denoise data. You may have noticed one of the listed data sets was called fix cleaned. And what that means is that it was trained using a machine learning classifier to identify things like movement artifacts, physiological artifacts, other kinds of noise from the scanner, filter out from the data so that what you're left with is a cleaned data set. So for those who are interested, Fix is a software package developed through FSL, one of the main fMRI analysis packages. And here's a brief overview of what it looks like, in which you do an independent components analysis. You decompose both the spatial and the functional components. You get these maps, you see where those components load for the most part, and then you, as the user, make an educated guess as to whether this is noise or signal. So here, since a lot of the loading is on the rim of the brain, and there's no apparent structure to it in the gray matter, I would probably guess that this is a noise component reflecting motion. Also notice that in the time series here, there's a large spike, which probably represents that the person was moving their head at that time. Now this component, on the other hand, has a little bit more structure. It's bilateral, it's mostly within the gray matter, and I'm more inclined to believe that this represents a signal component. And also notice here in the power spectrum, there's a greater amount of lower frequencies, which again probably indicates that this component is related to signal. Now, just in my own analyses, looking at data from the Human Connectome project, I looked at analyzing this through a typical FSL pipeline and then also using Fix as well, using a machine classifier that has been trained on the Human Connectome project. And here's just, within a random subsample, what the results might look like. So I can't say too much whether this improves the overall results. This is looking at a default mode network. Okay, so green is after I applied the Fix filter. Red is just a typical FSL processing pipeline. Maybe it reduces some false positives. Maybe it cleans it up a little bit. Just showing you some preliminary analyses that I've been working on with the Human Connectome project. Yeah. So for those who also want kind of a more accessible introduction to using some of these, there have been a lot of advances in the software packages that we typically use, at least in my field, for things like resting state connectivity and task-based analysis. So I'll show you a very useful feature, especially if you're trying to just get your feet wet or trying to train some of your graduate students, for example. And one of these is through the CONN Toolbox, a very popular resting state analysis and general connectivity package. So I'm going to give you a brief demonstration here on my own machine. So through the CONN Toolbox, you'll notice that you are now these days, as far as I think a year ago in their latest versions, you can directly access things from the 1000 Functional Connectomes project, one of those databases I mentioned earlier. download everything and pre-process it in one go. So with minimal intervention, you can download any of these satellite sites and download the images and pre-process them. So what this would look like, and here the CONN Toolbox is built on MATLAB, for example, if you're curious about it. If I open up the CONN Toolbox, just give it 1 second. Typically what you would do is you can just open up a new project. You could load your data, start analyzing it. But one of the most recent advances through the CONN Toolbox, if it's your first time doing it, you could go to Project. And then one of the new features is you go to New Download, and then from one of these public available data sets. FCP referring to the Functional Connectomes project. You just specify what do you want to call the project file name. I'll just use the default for now. And then it will give you a list of all the different possible data sets that you can download. So things from Ann Arbor, for example, hey, that's where we live, to Beijing, to Dallas, and so on. Just for demonstration purposes, I'm going to select the smallest one with an N of 8, because that should take relatively little amount of time. And it will give you the option to both download and pre-process. So CONN will do all this filtering, all this pre-processing, realignment, normalization, and so on. Or you can just download only, which is what I'll do for right now. So in the background, what's happening? It's connecting to where this database is. It's going to download this type A B data set, download it, unzip it, and then load it into your CONN Toolbox. So at the end of the day, it'll look something like this. Click on structural, for example, and take a look at all of these different structural images that have been uploaded. All right, so at this point, again, if you're familiar with the CONN Toolbox, and if you want to learn more, I have some tutorials about how to do it. You could go through pre-processing. You could use some of the demographic information available as well. I believe they also give you some of that too. And then just process that as you would a typical data set. So again, this is to show you some of the advances in the software packages that have made it a lot easier to interact with some of these online databases. Now, one of the last ones that I'll show you, I think this is the last one actually, is something called OpenNeuro. Again, more accessible, a little bit easier for people who are new to this. And OpenNeuro is different from some of the previous ones that I showed you. So things like the Human Connectome project, things like ADNI, for example. OpenNeuro is different in that a lot of its data is stored in BIDS format. So this is a new trend in the field of neuroimaging, which makes it a lot easier and more interoperable to take data from one set of people, one group, and to easily analyze it by another group of people. So BIDS' brain imaging data structure is a standardized way of organizing functional and anatomical data. And like I said, this makes it a lot easier to just make very tiny edits to scripts, but then it should work with a new data set or to replicate the same data set much easier than it did before BIDS came along. With OpenNeuro, you can download the data with Amazon Web Services or something called DataLad. And I'll give you a brief overview of what this website looks like. So OpenNeuro, make sure I have it here. Yes, simply openneuro.org. I use this all the time, especially for my course, Sci 808, and also if I ever give workshops, I find OpenNeuro very, very useful. So you'll notice they have about 50,000 subjects, over 1,000 public data sets, and they encourage people who publish their data in a journal to also upload it to OpenNeuro. Because now it's in a standardized format, people can easily grab it, download it, compare their results, and maybe even do novel results as well. You'll notice they have many different modalities: MRI, PET, EEG, MEG. They're also adding things like arterial spin labeling, diffusion imaging, for example. For now, I'm going to show you a classic data set that I like to work with. If I just enter a keyword, flanker, it's a very common psychological task to measure things like executive function and inhibition. So if I go to flanker task, event related, give you an overview of what this looks like. Let's say that you published the flanker task, some fMRI study of the flanker task. Where do you see related bold signal in the brain in response to the flanker task? So when somebody uploads their data, they put it into this so-called BIDS format, which again is very, very useful. It's a relatively new trend, which I think is going to help advance the field immensely. So notice every subject has a unique identifier starting from sub-01 to however many subjects you have. And then subfolders like ANAT and FUNC, storing things like your anatomical data, for example, and functional data as well. It's very easy within the web browser if you want to do a quick eyeball QA check. They have a Papaya viewer, which makes it very, very simple to do a very brief overview of the data. If you want to download everything, you can download individual files by themselves. Or what I find more useful, if you... Oh, sorry. Oh, I thought I heard something. Or you could go to download and use, like I said, Amazon Web Services. So what this would look like if I simply copy and paste it. I'm not going to wait for the whole thing because it does take a while. But just to show you kind of how simple the interface is and how this downloads the entire data set. So I've already downloaded it. I'm going to stop that for now. I've already downloaded it, put it in this flanker data set, and now this is within my desktop. And at this point, I can start to analyze it however I want. Something else I want to point out as I'm going through all of this is I do have an ebook if you're curious about how to start working with some of these data sets. I do have an ebook that walks you through how to analyze things like the flanker data set using, it could be SPM, AFNI, FSL. Also, if you want to use the CONN Toolbox to analyze that functional connectome data, there are walkthroughs for that as well. So again, after this meeting is over, you're curious, want to learn more, obviously you can get in touch with me or simply go to Andy's Brain Book and look through whichever software package you're most interested in. I'm currently working on things like the Human Connectome project. Let me see if I have this down here. Yeah, Human Connectome project. Still under construction, but I'm hoping to give a very useful introduction how to download one of these larger data sets, process it, and start to work with it on your own, as well as how to interpret the results. So that's an overview of OpenNeuro and also some educational resources to continue working with it as well. Now to end, I just have a few more slides, but I want to talk about some of the issues with large databases as well. This has been, again, relatively superficial, just giving you a survey of some of the major players like Human Connectome Project, UK Biobank, and others as well. Now, like I said, the typical fMRI study compared to these large databases is minuscule. We're talking about around 25 subjects compared to thousands, maybe tens of thousands of subjects. But I want to also vouch for and champion these smaller data sets It's not as though they're completely outdated. It's not as though the results are not interpretable or invalid. And I also want to point out that large databases have their issues as well. Number one, which I alluded to with the ADNI database, is that data can be collected from different sites and also from different time periods. So for example, with ADNI, there are changes in acquisition protocols over time. The very first phases of ADNI were maybe 10, 15 years ago. And obviously scanner technology changes, it becomes better. And the subjects they're acquiring these days, they're probably not going to use the same scan sequences as they did 15 years ago. So those can change. And data quality can change too. Like I said, scanner harmonization is an active field of study. There's still no simple way that I know of to account for the fact that data is acquired from different sites and also different time points. Really, the simplest and coarsest way to do it is to simply add scanner site and maybe scanner phase as a second level covariate to account for that variance, which again, not everybody agrees with that. And there are many different nuanced ways to account for it, which is an ongoing field of study. So that's one potential issue. Something else I also want to point out, this is a study by Scott Merrick and colleagues published 2 years ago in Nature Neuroscience. And this is a very dense, complicated figure. But what I want you to focus on is the fact that they used, I believe, UK Biobank. They used many thousands of subjects. And they looked at things like cognitive ability and also psychopathology, which I forget exactly what this was, but maybe things like anxiety, depression, so on and so forth. Essentially, can you predict any of the variability in things like cognitive ability or psychopathology using things like structural measurements or resting state functional connectivity? And you can see in these plots, in this third row here, the effect sizes, the correlations, let's roughly equate those with effect sizes, is somewhere around like 0.1 to 0.2, even across many thousands of subjects, looking across cognitive ability and psychopathology. And their key point, if you look at this figure down here, is that if you take a random subsample of maybe 25 subjects from this larger database, you can get completely opposite correlations of things like resting state connectivity and cognitive ability. So that suggests if we're just looking at things like behavioral metrics or other phenotypes, trying to establish correlations with a typical sized fMRI study is probably not going to cut it. And Katerina Gratton, I thought, wrote a very insightful reply to all of this after the Merrick paper was published. She agreed, based on these data, that cross-sectional studies with a very small n, let's say 30 subjects, are useless or probably going to give you very spurious correlation results if you decide to use them. Also, these consortia studies like UK Biobank, ABCD, as we saw in the previous slide, they may have small effect sizes, true, but they can be comparable to other small effect sizes in the field of public health that are still useful, like things like lead exposure and cognitive development. Again, the effect sizes were pretty small, but still significant, still had some kind of measurable impact on people's development, which led to public policy changes about building codes, for example. But one of the problems with these larger databases is that they usually avoid novel experimental questions and designs. So imagine you've collected 10,000 subjects, and then a new scanner sequence comes that seems to make your previous one obsolete. Now, turning it is like turning a gigantic ship. It's a very slow process, can be very difficult, maybe impossible to do. So how do you accommodate rapid changes in the field? That's more something that can be done with a smaller study, for example. So larger consortia have their place. They can be very useful, but they also have some disadvantages by virtue of their size. It's a double-edged sword. So here's the figure that she used to show the distinction between them. So focused studies, even though they have a small n, if they're very focused and well controlled and have a good experimental design, you can still get very high signal to noise ratio. And they can be very useful for things like precision medicine, guiding clinical care. Consortium studies, they can be extremely useful. In fact, they're necessary for measuring more population level variability and giving you better information about population tendencies and things like providing guidelines for policies. And another thing I want to point out, which is again very important to keep in mind, is many people who are junior researchers, they may be limited in funds and in opportunities, and I don't want them to be discouraged by some of these larger database results from carrying out their own studies. It's just a matter of what exactly are you looking for? What kind of claims are you trying to make? And like Gratton said, smaller studies can yield useful results through higher signal and lower noise designs. Things like within subject longitudinal studies can be also very, very useful at controlling for many different types of variability. And so one of my last slides is one of those examples. This is from Dylan Newbold and colleagues in 2020, looking at only 3 subjects where these again, subjects that had no kind of physical disability, but they put a cast on their arm anyway to immobilize it over a period of weeks or several days. And you can notice things like, obviously, things like grip strength goes down in that particular arm and hand. The functional connectivity before the cast goes on, the correlation between motor cortices, virtually disappears. It's really not detectable at normal thresholds, but it reappears once the cast is taken off. And you can also see that during the cast phase, there appear to be these random pulses, these random establishments of functional connectivity between the hemispheres that kind of go in and out of existence. They kind of appear at random. And this can lead into some insight as though even though the cast has immobilized their arm, there's still like these radio pulses from one part of the brain saying, has this been reconnected? Can we still use it? It's still immobilized. Okay, we're going to ignore it for a little bit, but still put out some feelers from time to time to try to reestablish connectivity, which could provide some insight into things like, prosthetics, brain behavior interfaces. How can we make this better? Okay, so to summarize, I gave you a sketch of some of the different resources that you can use for different resource questions. Things like ADNI for Alzheimer's subjects, for example, or I should say subjects with dementia. The ABCD study, if you're interested in development over the adolescent phase. And also things like Open Neuro and the Functional Connectomes project, which are more accessible and a little bit easier for people to just get started with larger databases if they want to. Be aware of prerequisites, privacy policies, for example. Again, with things like ABCD, things like ADNI, obviously they're much more concerned about privacy. You should be too, so just take that into account before you apply for access. And lastly, as I referenced with the ebook, any questions, just ask. My job is to provide resources, educational materials, and I hope that anybody who's found this interesting is then motivated to pursue this further, and I'm happy to help with that. So thank you for your time. At this point, I would love to take any questions. And thank you, Shruti. I appreciate that very much. Yeah, I tried to make this as relevant as possible for people who are going to be using these kinds of resources. So thank you again for your time, and I want to open this up for any questions in the time we have left.
Thank you very much. That was really a very thorough and comprehensive talk, and virtual round of applause to you.
Thank you.
Comments or questions from anyone? So,
question. Sorry.
Go ahead.
Yeah.
Gary, can I go ahead?
Please, absolutely.
Thank you very much. Excellent talk, and thank you for making us aware of these resources. I guess my question was that from the clinical standpoint, and I'm a clinical neuroradiologist who thinks about different diseases, my question was, in these large data sets that exist, how much granularity is there about their underlying disease processes or lack thereof in the subjects who have been scanned?
Right. The question was, how much granularity is there in these different kind of clinical diagnoses? And I can't say too much about that. I'm not entirely sure. I do know that, for example, with the ADNI data set, they try to give different levels of severity as far as cognitive impairment, but they are pretty coarse, for example. That's a clinical data set I'm most familiar with. So, and for some of these larger consortia databases, they do have relatively, say, coarse levels of granularity when distinguishing between some of these different subgroups, as far as I'm aware of.
So I guess that would be another advantage of those small number focused studies, that we can explore some very specific question and very specific patient population, which you cannot with this large data set.
Exactly. That's the trade-off. If you have your own study, you have so much more control over who's in it, how it's designed, what you can measure. Mm-hmm.
Thank you.
Yes, thank you for that question.
And Dasha put a question in the chat about, are these Linux-based?
Yes. So some of the resources and tools, they're not necessarily Linux-based. I did show a command line to demonstrate how I would download and analyze one of them. So for example, openneuro.org, they do, I would say they assume that you have some familiarity with Linux, because things like AWS and also DataLad are run through the terminal. But things like Human Connectome project, you don't necessarily need any kind of command line. And like I said, they have that workbench graphical user interface for a lot of both visualization, but I'm not so much sure about analysis. In any case, I would say that if you are planning to do any kind of further analyses, you do need to have some familiarity these days with Linux. MATLAB is the other alternative, which also provides access to things like the CONN Toolbox, downloading some different data sets as well. But I would say Linux, some familiarity, some fluency with it is highly recommended.
Any other questions?
This is Lumir Hajitsky. Great presentation, very, very interesting for the big data sets. One question actually, for the imaging part of the data sets, do you have kind of annotations of the anatomical location of the abnormality, disease, or something like that, or everything is more or less based on the analysis of reports and is more or less kind of macro information?
I would say more the second one. Again, so this is the thing. I'm not so much a clinical imager. I'm much more on the research side of things. I do have some contact. Let's say, for example, the Alzheimer's Disease Center here at Michigan. I help them out sometimes. So I can't say too much about that. And I'm hoping that, obviously, as people are aware of these, they can use them for specific purposes without having to collect their own data sets, for example. So that's what I would say about that.
Great. Thank you very much.
Thank you.
Okay, thank you all very much for participating. And again, thank you, Andrew, for great presentations. Have a great rest of your day.
Yes, thank you all so much for your time. And thanks again for inviting me to do this. I appreciate it.
Thank you very much.
All right. Thank you, everyone.